Okay, good afternoon, everyone. My name is Carl Adams from St. Francis Xavier University, and I am the Atlantic Regional Coordinator for the 2022 Canadian Association of Physicists Lecture Tour. Before we get started, I have a couple of announcements about how the talk will proceed. All questions will be handled at the conclusion of the speaker's presentation. Please post your questions in the Zoom chat at any time. The questions will be posed anonymously to the speaker by the MC. All students participating today are encouraged to remain after the talk and participate in a 30-minute breakout informal discussion with the speaker. We also invite any faculty or other attendees from the host institution to participate as well. Now, it gives me great pleasure to introduce our host for today's CAP lecture tour talk, Professor Greg Christian from St. Mary's University, who will introduce our speaker.
Thank you, Carl, for the introduction. So welcome everyone to the sixth and final talk in the CAP lecture tour series for 2022. My name is Greg Christian, and I am pleased to host this talk from the Department of Astronomy and Physics at St. Mary's University. We thank Science Atlantic for sponsoring this talk. We had hoped to be able to have this talk in person at St. Mary's and stream across Canada, but with the rising COVID-19 cases in January, the decision was made to have this lecture tour purely online. The lecture tour is sponsored by the CAP Foundation and the Canadian Association of Physicists, a national network of about 1,700 physicists working at universities, companies, and research institutions across Canada. The CAP works on behalf of Canadian physicists to support physics education and research, and sponsors a number of physics activities, including this lecture tour and national research conferences. The CAP also delivers a number of programs for undergraduate and graduate students, research conferences, physics scholarships and prizes, and a student advisory council. If you have not already joined CAP, please consider becoming a part of Canada's largest network of physicists and physics students. It's free for undergraduate students, and the first year of membership is free for graduate students as well. Today's talk will be 45 minutes, followed by 10 minutes for questions. If you would like to pose a question to the speaker, you can type your question in the chat at any time, and one of the moderators will ask the question at the end of the talk. Following the talk today, you will have the option to join a breakout room where you can speak directly with the speaker, ask additional questions about the research or about related career paths. And now it gives me great pleasure to introduce today's speaker, Professor Ozzy Mermut from the Department of Physics and Astronomy at York University. Today, Professor Mermut will present her talk entitled Biophotonics, Shedding Light on Biosensory Disorders and Age-Related Degenerative Diseases. And welcome, Professor Mermut.
Thank you very much, Greg. Let me see if I can start sharing my screen as we normally do. Hopefully everyone can see my screen. I'd like to first start with a thank you to CAP for organizing this amazing CAP 2022 lecture tour. Merci pour l'introduction et l'Université d'accueil à St. Mary's. And I'm really excited here to talk to you about some progress in my lab related to combining light with life sciences to figure out what's going on with age-related degenerative diseases in our biosensory systems. And so I'll get started with that. But first, I'd like to start with a land acknowledgement. York University recognizes that many Indigenous nations have long-standing relationships with the territories upon which York U campuses are located. that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, and the Huron-Wendat. It is now home to many First Nation, Inuit, and Métis communities. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. And you can see some pictures here from York University across campus in this beautiful land, which we share, and the Center for Indigenous Studies here at York University, and various wildlife and beautiful trees that surround this beautiful campus. I'd also like to take a moment to resonate CAP's message regarding the war and oppression and persecution we are witnessing at this time in Ukraine. Let's please continue to show our strongest support and solidarity to our scientific colleagues and civilians in Ukraine. I want to also first start my lecture, hopefully by doing the most important thing, I think, which is to thank the wonderful students and postdocs in my lab that do all the amazing work that I'm going to be presenting here today. So thank you, students, for being so interested in this field and bringing this field to its frontiers. I also want to thank my students in my biophysics courses, particularly those in the 4,000 and 5,000 level, who have eagerly jumped to volunteering their brains and other organs for pilot data, with ethics, of course, as volunteers for a variety of different biophysics demonstrations. And you see a few of these examples from my biophysics courses, and students getting really involved in some of these techniques that we are going to be discussing today. And I also would like to do my acknowledgements now, particularly my wonderful interdisciplinary team of collaborators across various universities in Canada and across institutions in Canada, and also internationally. I couldn't have done this work without their wonderful collaboration that really brings the frontiers of biophysics research to its helm. And I'd also like to thank the funding sources for the research conducted in my lab and my various collaborators at the National Optics Institute, INO. And I'm going to apologize also right now if I forget to credit any images here in this presentation. All right, so in our lab, we're interested in understanding what happens to biosensory systems as we begin to age, and particularly dysfunctions that lead to degenerative diseases, from diseases of vision to neurological disorders and to cancer. And today I'm going to focus mostly on vision diseases, and hopefully perhaps in the discussion we can talk about some work that we're doing towards applying biophotonics to neurological disorders and our understanding of how to treat them more effectively. And so today I will focus here, and hopefully we can get started by first talking about why vision disease is such an important problem to solve. This is an article from CTV, and it discusses not only the huge economic costs associated with vision loss to Canadians, but it also highlights the importance of what happens when we have debilitating pathologies of our eyes and our vision. And so one of the important things is that you can imagine if you have vision loss. This is a case for of someone who has age-related macular degeneration. You can see this is what an individual who has AMD sees, which is not very much. And it affects your ability to be able to perform everyday activities. And the inability to perform these daily activities has been noted with increased cases of depression associated with vision loss. So this is a real big problem. It's not just our vision loss, but it's our ability to be able to do our everyday normal tasks. In looking at visual dysfunction as a whole, there are about a billion people who suffer from visual dysfunction. And in fact, AMD is one of the most common forms of diseases related to aging of the eye. That's age-related macular degeneration, and affects nearly 200 million people worldwide above the age of 60 years. And it accounts for about 8.7% of all blindness cases worldwide, and particularly affects those in developing countries. I don't need to go through the economics of vision loss, but we do know that there's a lot of money that is spent both in Canada and the US attributed to managing vision loss and blindness. And in fact, this has been one of the major incentives for developing this area and new ways of probing vision loss and treating vision loss. In fact, in some journals of ophthalmology, it has been reported, in fact, that vision loss can be worse than the loss of a limb, memory, hearing, speak, or even contracting HIV. And some people have equated it to Parkinson's disease and heart disease in terms of the fear of individuals losing their vision. So along comes the coronavirus, and those living with visual dysfunctions have been exasperated. The situation has really been exasperated, particularly for those with visual disabilities during the pandemic. And recent studies actually have shown that our perception can be severely hampered by our inability to visualize faces. And so my colleagues at the Center for Vision Research at York have shown some really interesting studies where, in fact, wearing masks can affect the visual performance in identifying and perceiving faces of individuals. This is vital to our daily function. So you can imagine that this is particularly problematic for those who are suffering with vision loss at this time of the pandemic. Even worse news. COVID-19 has been said to induce accelerated vision loss by direct invasion of the retina, which has been reported to be affiliated with inflammation, and this inflammation has been shown to have optic nerve damage. And so not only is vision loss traumatic on those folks that are living with it and our ability, again, to perform everyday tasks, but COVID-19 has really exacerbated this situation. So I think it's long time that we figure out how to better manage these diseases. And so if we look at our various biosensory systems, I've highlighted a few of the areas of our research here. There are some commonalities with respect to aging and age-related biomarkers for degenerative diseases of the eye, the brain, and various cancers. And so let us examine some of these things by these common markers for aging and disease. So if we look at something like Alzheimer's and cancer and maculopathy, we observe some of these continuum of common attributes or biomarkers related to these pathologies. Things like inflammatory response, signaling, changes to a metabolic function, and oxidative stress that occurs in the cellular function, cellular senescence. All of these are common attributes with respect to these diseases that we're interested in studying. We're also really interested in looking at not only these functional biomarkers, but structural biomarkers. And as we age, the tissue architecture and structure, such as the collagen and elastin molecules in there that are in our vasculature, change. And this results in change in endothelial tissue and its elasticity over time. So again, when we're looking at aging and age-related degenerative diseases, we want to look at both structure and functional biomarkers that are linked to these things. So let's take a look at this case with age-related macular degeneration. So we have this image of the eye and the retina here, which has been expanded here. And you can see this beautiful 11, well, you can't see it here, but you can identify it in these images here. But you can see how you have this beautiful architecture of the retina. It has 11 layers to it in the human retina. And when we have age-related macular degeneration, this is what happens is that we have neovascularization and hemorrhaging that perturbs this Bruch's membrane, and fibrosis, which occurs in this degenerative disease that essentially perturbs this architecture of the retina that is responsible for photoreception. And so these hallmarks have been able to be visualized only recently due to the advent of optical coherence tomography. This is an interference-based measurement, and OCT involves taking light and looking at the interference between sample tissue and a reference arm, and looking at the interference pattern produced with that construction of an OCT image. So as you can see, this is a beautiful, healthy retina that we've characterized here, and this is an unhealthy retina that has a variety of different perturbations to the Bruch's membrane, and eventually results in neovascularization. And so it is because of these arising biophotonic techniques that we are able to see structures of the eye that we were never able to see before with exquisite resolution, both axially and laterally. And so why am I here today? I think I'm hoping that I can express to you the importance of the role of biophotonics in addressing age-related conditions. And biophotonics comes from the Greek words bios and phos, which combine light and life. And we do this to address these conditions and look at various ways that we can target various forms of treatment, as well as identify these at an early stage. And why is biophotonics such a beautiful approach, in my opinion? It comes from its exquisite ability to have particularly high sensitivity coupled with specificity, where we can also obtain these images with extremely high spatial resolution. So it's a really amazing way that we can probe the cellular, molecular, and tissue functions in the body, compared to a variety of other techniques, also very important in the role of treating these diseases. But again, unlike some of these other areas, we can combine molecular specificity with a great deal of spatial resolution. And so this gives us various advantages for being able to detect biomarkers of aging early on. And if we can detect these early on, perhaps this can give us some insight into what we can do in terms of disease prevention. And early detection almost always is linked to better treatment, more targeted and personalized treatment of the disease, as the disease expresses itself in a variety of different forms in different individuals. So perhaps one of the most important roles of biophotonics has come from the development of advanced laser technologies. And this is evidenced by the 2019 Nobel Prize winner in physics, Donna Strickland from the University of Waterloo. And I was at her Nobel Prize talk at CAP at 2019, and she indicated numerous times the importance and impact of lasers in medicine, and particularly in eye surgery. And so lasers, particularly fast, ultra high speed, ultra high power lasers, are really driving this field, not only in ophthalmology, but a variety of different photonic treatments in cancer. And so I think this is a really timely pursuit to solve these important challenges. So perhaps taking a step back 100 years before I got interested in photons, and I really am, I've been interested in photons ever since I can remember, but hundreds of years before me, some greats also asked questions about light and matter. And photons are still a big mystery to me, and it amazes me the complexity, in fact, of photonics every day in our research activities that we do. And Albert Einstein dedicated his life to understanding and determining what exactly a photon is and what it does. So I really think it's important to spend a little bit of time getting back to basics here and the role of biophotonics through understanding how light interacts with matter, including biological matter. So in the 1600s, late 1600s, Newton argued that the very geometric nature of light, its laws of reflection and refraction can only be explained if light behaved as a particle. And the beautiful experiments of Thomas Young thereafter in the early 1800s, particularly the interference experiment where light is sent, light source, coherent light source is sent through a double slit, and we can observe these interference patterns that show up on a screen, demonstrated, in fact, the wave nature of light. And so the very basic nature of light has been under debate for centuries. And later on in the 19th century, there's been more cases of wave-like behavior, but also particle observations. And really in the early 20th century, we have Albert Einstein with his photoelectric effect, and Max Planck demonstrating energy quantization, really revolutionizing the definition of a quantum packet of energy through quantum mechanics and defining a notion of what is a photon. And so we've now convinced ourselves that light at this point behaves as a dual citizen, having both particle type properties and wave-like behavior. And so we have light behaving as a wave, and the speed of light being dependent on the wavelength and its frequency. And as we have light that is moving through air, we're going to call that refractive index 1 for simplicity. It will slow down as it encounters different types of material, going through the cornea, the lens, various aqueous humor, and hitting the back of the retina. And of course, this interaction results in the change of speed of light in these various media, so that we can look at what happens to the photons at the back of the retina. And each particle, or photon, we're going to call it, has a discrete, or the molecules from atoms and molecules have discrete energy levels that are defined by our well-known E equals h nu. And so we can exploit the energy quantization of atoms and molecules such that when we're doing things like shining light, near infrared light, onto the brain and looking at the vasculature, in other words, the hemoglobin in the brain, we can capitalize on the absorption behavior of these hemoglobin molecules for doing excitation and emission spectroscopy at various discrete wavelengths and various discrete energies. And of course, we've been doing this for quite some time, and quantum mechanics has given us these energies for much more simple atoms like hydrogen. And what we're interested in, of course, is looking at more complicated biological systems, things like myoglobin, which are essentially oxygen binding proteins that are in muscles, and looking at their spectral behavior under different conditions. And so we have this beautiful electromagnetic radiation spectrum that includes long radio waves right up to very short waves in the X-rays here. And we can exploit that entire range in biomedicine. What I'm going to talk about today is really in the visible and near infrared. And there's a reason for why we're interested in this, because we're able to probe the various tissues with non-ionizing radiation, and radiation that can penetrate deep through the tissue. And depending on the energy and the frequency of light that's used to probe these tissues, we can get a variety of different interactions, and allows us to do things like rotational, vibrational, and electronic spectroscopy on these systems. And it really comes down to the quantization of light that we can attribute a variety of different advances in biophysics and biomedicine. So this is a beautiful image of a neural culture that's been stained with a Bornavirus, and as well as a tetanus that looks for a tetanus toxin. And we're able to do things like surgery with lasers, never possible before. And also, due to the very fundamental nature of these light matter interactions, we can, through photoelectric effect and Compton scattering, we're able to do diagnostic X-rays as well as therapeutic X-ray treatments. All right, so let's talk a little bit about what's happening here. So when we have some, let's say, hunk of tissue here, this brain material, and we send a collimated beam of light through a collimator and expose that light to the tissue, a number of different things can happen. One, you can already guess, these photons are going to collide with the molecules and atoms in the tissue. Another thing that can happen is absorption. So it absorbs with a discrete energy, and the light is absorbed and it can emit thereafter photons either through radiative or non-radiative processes. And then we can use our detector to look at the transmitted light from these photons that are completely transmitted through this specimen. And all of these processes are really governed by quantum mechanical selection rules. And so if we want to look at how light interacts with matter, and in particular biological matter, we need to develop a model for looking at light matter interaction. And so here again, we have our slab of brain, and we bring upon an incident light. And what happens is that the incident light then decays as it passes through in an exponential fashion through this tissue. And this is encompassed with the Lambert Law that's expressed here, which is a function of the cross-section of the material, or in other words, of the linear attenuation coefficient and the distance of the light penetration. So we have this very important relationship that encompasses how light interacts with matter as it goes through the tissue, as I showed you in that case with the OCT and those retinal layers that we saw. Okay, so what is this attenuation coefficient? This attenuation coefficient is broken down into absorption and scattering, so mu A and mu S scattering coefficients. But it's a little more complicated than that, and I don't really have time to get into it today, but there are several mechanisms that are implicated in this attenuation coefficient that I've showed here on the right-hand side. And one thing I really want to express here is, and one of the beautiful features of light is is the wavelength dependence of this equation, the Lambert Law. So we really have a wavelength, the intensity decaying is a function of the wavelength of the light that you're using. And so what I'd like to do, and it's shown here in case this doesn't work on the Zoom, here's a picture on the right-hand side of a 5 milliwatt green laser and an even less powerful 3 milliwatt red laser. And so I'm going to try to do this. Let's see if it works. All right, so hopefully you can see the light that's passing right through my thumb. Okay, hopefully you can see that. If I do the same thing with a blue laser pointer, hopefully you can see this is nice blue laser. It tells me that this is at 405. Same power, you can see that there's nothing going through, okay? And this all comes down to the wavelength dependence of the light penetration through the tissue. In other words, blue light is more attenuated in tissues, in many tissues, more so than longer wavelengths of red light and infrared light. And so we can take advantage of this for probing a variety of different types of chromophores in the tissues, as you can see, and do different forms of spectroscopy for where we can absorb different types of chromophores that we're interested in. Okay, so that's really great. But one of the important tasks, I think, in the biophotonics community is to develop quantitative metrology techniques to ascertain these optical properties, namely the absorption and scattering behavior of tissues. And in this effort, phantoms, which are essentially just simulants of optical simulants of tissue, okay, ones we can set the absorption and the scattering behavior of. These phantoms have been developed over the last, let's say, about 10 years, 20 years perhaps, in order to be able to do more quantitative and systematic studies of the absorption and scattering characteristics of biological tissue. And with my collaborators at INO, we've looked at time-resolved methods, single photon counting methods, where we can send a pulse of light and look at the temporal point spread function as the photon migrates through the tissue. And by coming back to the Beer-Lambert law, or a form of the Beer-Lambert law, we're able to extract very quantitatively the scattering and absorption properties of these tissues. And so you can imagine this allows us a way of being able to systematically study tissues as precursors to studying real biological systems in a quantitative fashion. Okay, so I've talked to you about the fundamentals of light-matter interaction and how we can interrogate our various biosensory systems from the eye, and it turns out the eye is a really interesting connection to the brain. So what I'm telling you for the eye, I'd like you to also think about how we can use the eye as a window into the brain. And again, we can talk about that further in the discussion after this. So I'm not the first one to do this, obviously. Lasers have been around for 50 years, or well over 50 years, in fact, for the treatment of a variety of different diseases for the eye, for age-related macular degeneration, diabetic retinopathy, retinal vein occlusion, and cancers of the eye like retinoblastoma. But it's only more recently that we've begun to develop more appropriate, I would say, techniques for being able to deal with really treating the lesion and not the healthy tissue. So of course we have wonderful photoreceptors in our eye here, and when we're treating the disease, we want to minimally affect those healthy photoreceptors. And so this has brought upon the incentive to develop targeted types of treatment. So the conventional type of treatment that's being used for treating especially diabetic retinopathy or any kind of vascular hemorrhaging in the eye is photocoagulation. And photocoagulation is simply, I guess in its most simplest sense, a thermal-based mechanism where it cauterizes those leaky blood vessels. Okay, so these are those spots that you see after a photocoagulation laser has been implemented in the retina. And what you see also here is a lot of photoreceptor damage, which is irreversible. Okay, so we've managed to restrict the disease and the hemorrhaging associated with the retinopathy, but we've also killed a lot of healthy photoreceptor cells in here. So there's been a great deal of effort in the last 10 years to look at selective ways of treating the retina, and I will be talking about that today. But here's an image of the same retina that's been treated, however, with selective retinal therapy, and you see very, very little collateral damage to the healthy photoreceptors. I'll also be talking about photodynamic therapy today, and the basis of photodynamic therapy is that we use essentially light-sensitive drugs that selectively uptake in tissues, and only upon illumination of the tissue containing this photosensitizer drug, we are able to generate, or convert, I should say, molecular oxygen to singlet oxygen. And that singlet oxygen, where the photosensitizer is namely the tumor, is able to kill the cells in that region of interest. So you can see there's a lot of different types of therapies that are available that hopefully are able to better target the tissue lesions as opposed to the healthy tissue. And so I'm going to talk about some of these and some of the challenges associated, in fact, with biophotonics. We've already talked about the fact that we can't get through as deep through tissue with blue light, for example, compared to red light, and certainly not as deep as some other imaging modalities like CT and MR. So we do have limitations in terms of the depth of penetration. The dosimetry also becomes really complex, and hopefully I'll give you a snippet of that later on. So we want to be able to measure how much energy we've actually delivered to each individual's tissue in an accurate sense, so that we're not giving excessive energy and mitigate the collateral damage to the healthy tissue. And I will also be talking a little bit about our work related to addressing the complexity of the models that we use, particularly the retinal pigment epithelial layer. So let's start with this. We want to treat the eye. We have some visual disease and we want to treat the eye. How do we achieve selective targeting? So here's an OCT image of the eye. Okay, and this is a nice healthy eye. So again, we can do all sorts of interesting diagnostics, but once we see some degeneration here, at a certain point, ophthalmologists will intervene with treatment. And one of these forms of treatment is laser treatment. And in untreated tissue, this is a sample right here. This is what it looks like. So if we took the RPE layer here, that's the blue layer that you see, the retinal pigment epithelial layer, and it's shown here as the untreated tissue. If we expose it to some radiation that's not targeted, you see all these little red dots are cell death indicators. Selective treatment allows us to be able to target much more specifically the damage that we can apply to the diseased lesion. And I probably don't need to talk too much about this, but all of this comes back to modulating where we're sitting in terms of the laser pulse duration and the power density that we are giving to the tissue. And that will affect what region of the interaction, light matter interaction range we are dealing with. So for example, when we're talking about high irradiance and very short pulses, we are talking about doing photo disruption and ablation type of mechanisms. Whereas if we're sitting in much longer pulses, much less power density, we are interacting with the material through photochemistry. Okay, so we can modulate the radiance and pulse duration for different mechanistic purposes of how we want the light to interact with the matter in a selective way. So selective retinal therapy is kind of the counterpart to the traditional therapy of that is predominantly photocoagulation based. And typically in photocoagulation, we're dealing with pulse durations that are greater than 100 microseconds, okay? So this is an image, again, of what happens, histopathology image, what happens to the photoreceptors when we're dealing with much longer pulse durations, predominantly thermal means of thermal diffusion of the laser energy into the tissue. And because it's so diffusive, we're less able to control that interaction spatial confinement of that energy. However, if we start going into much shorter pulse ranges, so less than microseconds, okay, this is below the microsecond regime of laser pulses, we're able to now selectively confine, thermomechanically confine this laser energy to a much more spatially localized region in the retina. So selective retinal therapy has been a great deal of interest to many vision scientists as a way of selectively targeting lesions without leaving healthy cells damaged, which are, of course, vital to our vision. And so this is a really beautiful picture of what happens. It's a time-lapse photo of what happens when an excimer laser, a pulse of excimer laser, induces what we call a microcavitation vaporization event. And you can see here, this is a tapered laser, tapered fiber that delivers the energy. We have a bubble that's generated from this vaporization, and this microbubble then collapses. And that collapse and that local confinement of the microbubble generation is what allows us to be able to control the collateral damage to the areas of the tissue we do not want to have receive that laser energy. So here would be a case where we have very poor thermomechanical confinement, and this is a case where we'd have very good control of the confinement of these microbubbles that are responsible for the damage of the tissue. So I guess it's no surprise that new laser technology really is at the heart of exploiting these kinds of mechanisms. And here is a laser technology from the National Optics Institute. And what it does, it has this unique capability of being able to tailor the pulse format in such a way that we can vary the rate at which the energy is deposited. So this would be an example of a pulse that is a standard Q switch pulse. And here's something that we can do, which is an inverse, so to speak, Q switch pulse, where we're delivering this energy in a ramped style fashion, but in an inverse to a Q switch laser. And what we observe when we do this is that we get much better spatial temporal confinement of the laser energy, the thermomechanical confinement of the laser energy. And we see this based on the threshold ranges that we are able to induce a cavitation event. Okay, so the threshold becomes reduced by 30%. And we also see the cavitation control over the cavitation size of that microbubble with really tight level of control of the microcavitation event. And so what we're able to do is take advantage of shaping these laser pulses in such a way as to optimize that thermomechanical confinement. So this is really promising, and we think it's pretty darn cool. But how are we going to detect this in order to do an accurate dose measurement? It turns out it's not that easy. And so if you look at, for example, photocoagulation, the dosimetry is inherently there. It's visible, right? You can actually see the dots. You can see the vessels be cauterized, albeit in a very crude way and in a very thermally diffusive way. But we can visualize that that dose that's deposited and say, okay, we've done what we need to do in terms of sealing that vessel, that leaky vessel. But we don't have this ability when we're doing selective retinal therapy, mostly because these things are very short-lived. These cavitations are very fast and they're very short-lived, and they just cannot be assessed unless you use some really nice techniques like the one I showed you before to visualize this cavitation event. And so what we want to be able to do is be able to deliver enough energy to produce this this cavitation, this vaporized cavitation, and then stop right there, okay? So not deliver any more laser energy than we need to. And so we have this dosimetry problem. How do we do that? Oops. So the way that we're doing this in our lab, and again, in collaboration with the National Optics Institute, is to look at how we can use photoacoustics. So photoacoustics involves absorption of light, and this absorption of light generates heat and creates a pressure differential. And this in turn generates a sound wave. And so you can detect this sound wave at very high temporal resolution at acoustic frequencies of the cavitation in order to dose how much energy that we've delivered, such that we only deliver energy to the threshold that we require for cavitation. And so here is a photoacoustic spectrogram that we have of a laser pulse that's been delivered, and one where we're able to observe through photoacoustic imaging this vaporization event happening. So now we have this way of being able to dose effectively how much energy we're delivering to the tissue. Okay, my eye is different than your eye, which is different than someone else's eye. So we need to be able to target the various properties of each, everyone's individual eye characteristics in order to be able to appropriately dose what we're delivering. And these photoacoustic maps of the damage region match very well the photoacoustic response, that we're observing in the retinal tissue that we're treating. So they overlay quite well. Okay, so this is a very accurate way of dosing these tissues. Now, we haven't gone into clinical. These are all explants for the time being, but this is a really promising avenue, I think, in terms of dosing the energy in selective retinal therapy. Okay, so I'm going to take a little step back here and switch gears for a minute, and I want to talk about dosimetry in a little broader perspective, as our group has been doing more recently. We define dose as, laser energy delivered to an area of tissue. But in fact, we can really broaden this in terms of classical dosimetry techniques that are being used to looking at how we can do dosimetry from a molecular point of view. And my colleagues in medical radiation physics have long been talking about the importance of oxygen in dosimetry of various types of treatment. And in fact, the 2019 Nobel Prize for Physiology and Medicine went to these folks, and their discovery of how tumorigenic environments adapt to oxygen and the availability of oxygen. So we know oxygen plays a really important role as a biomarker in the development of cancer and tumor progression. And so we want to exploit this. We want to exploit this as a way we can also dose how much energy, light energy, that we are delivering to patients treated with these ailments. And one way of doing this is through a technique that we have developed in photodynamic therapy. Photodynamic therapy is, again, the type of therapy where a light-sensitive drug is injected into the system, and that light-sensitive drug locally uptakes where the lesion is. And upon photoexcitation, we have an interaction that occurs such that the photosensitizer transition goes into the triplet state and produces cytotoxic oxygen from otherwise benign and very well needed molecular oxygen. I'm not going to get into the pathway of these things, but you can see how complex these pathways are for generating the cytotoxic oxygen. And the fact that it's very multi-mechanistic in terms of the different pathways of creating cytotoxicity, either through radical ion means or through singlet oxygen creation. And so one of the things we've done in our lab is to look at how weak magnetic fields can be applied within the phototherapy context to sense the local oxygen concentration. And if we can extract the local oxygen concentration, we can figure out more about the mechanism and the role that oxygen is playing as we're treating. In other words, we can use oxygen as a way of dosing our photodynamic, very complicated photodynamic therapy treatment. And I'm happy to talk more about this again in the discussion, but this is all based on the principle of applying a weak magnetic field that induces a hyperfine coupling of energy states and splitting the degeneracy of the triplet state in these interactions, the photoexcitation interactions, and allowing for Zeeman splitting, that we're able to derive some information about the local microenvironment of the the treated tumor and its local oxygen environment. We're not the first ones to have done this. The concept of weak magnetic fields and looking at them in terms of radical oxygen species and singlet oxygen have been around for quite some time. Photosynthetic bacteria is affected by weak magnetic fields. We can exploit them to examine enzyme reactions. And even birds have a magnetoreception that is based on radical pairs that are produced in reactions that are very similar to photodynamic therapy. And so we think this is pretty cool stuff, and we wanted to demonstrate the use of weak magnetic fields and in the context of detecting these photosensitizers as we're treating these tumors. And so we've developed an apparatus to be able to do that. And what we can do is, in the presence of weak magnetic fields, we can detect the fluorescence lifetime behavior of the photosensitizer to examine what's happening in situ while the photodynamic therapy treatment is applied. And what we find is something quite interesting, that the lifetime of the decay of these photosensitizer fluorophores are extremely dependent on the oxygenation environment of these tumors. Okay, so here is a chick embryo, chorioallantoic membrane. It's been implanted with pancreatic cancer cells, and we treat them with photodynamic therapy in the presence of weak magnetic field, and we see that due to the Zeeman splitting and hyperfine coupling effects, we have an effect that tells us something about the local molecular oxygen environment. And if we calibrate this response, we can essentially get a molecular imaging map of the local oxygen concentration as we're treating the tissue. So hence we're looking at this as a very new way of being able to dose the treatment. I'm looking at the clock now, it's 2:53. So I'm going to leave off perhaps with this. And then I also had some more stuff to talk about, but I'll leave that again for the discussion segment. But I'd like to propose one other way of being able to look at how we can get some information about the local environment of tumors, be it for treatment or for looking at tumor progression. So one of the ways that we think is really promising is looking at markers, which are going to be biological markers that we can give to the tumor in a similar way that we do for phototherapy, but give them markers through which they can exhibit photosensitivity to oxygen when they're excited and based on their change of their conformation. So this is an azobenzene molecule, and it's a photo switching molecule. And when you give it light of the appropriate wavelength, it can switch from its trans conformation to a cis conformation. Okay, and like Mother Nature, we have molecules like azobenzene in our eye called retinal, and these are very important signaling molecules that are responsible for our vision. So our thought is, can we use the photo switching behavior of these molecules to sense the local environment of the tumor? So azobenzenes are really interesting for a variety of reasons, and I've thrown some of them up here. But I'm just going to leave you with the benefits of using these molecules in that they're optically tunable, so we can target the various regions of the spectrum, and they're also biocompatible. Right? They've been used in a lot of food dyes for quite some time, and so we have materials that are very biocompatible with tissues. And in our lab, we have been able to demonstrate that, in fact, the photo switching behavior of these molecules are very dependent on their microenvironment. In specific, we've shown that they are dependent on the pH of the local environment that this photo-induced switching is occurring. And more recently, Coral in our lab has looked at the oxygenation environment of these azobenzenes and modulating the oxygenation and noticing, in fact, that these things, the rates of these photo switching depend on the local concentration of the oxygen environment. And so these are promising results, but you can see that they're not all that sensitive. And so what our new work right now is focused on is attaching heme molecules here, okay, which are inherently able to capture, well, their whole job is to capture oxygen, and look at how we can sense the oxygen around this azobenzene molecule that's in the tumor by looking at how the rate of the photo switch attached to a heme molecule will change as a function of the local oxygen environment. So we're pretty excited about that. I think I will stop here. I wanted to talk to you a little bit more about the importance of how we model these things and create artificial models of the eye, but I think I've run out of time, and perhaps I will just forward this to the end here and save it for another time. So I hope I've been able to convince you that biophotonics is a really interesting area for probing age-related diseases of our biosensory systems. I've had a chance to talk to you about vision today. We're also doing really, I think, fun and cool work with the brain. Again, more for later, as well as cancer. And we're able to deliver more targeted therapies so that we're able to preserve our vision when we encounter these debilitating diseases. And of course, if we're able to deliver this targeted therapy, we need an effective way of dosing our treatment. And so I'm proposing to you that oxygen sensing is going to be the new wave of molecular level sensing of these therapies, many of which rely on the oxygen local environment of the tumor. And I can talk to you about some of the models that we're developing right now to emulate the aging retina with a lot of biorealism. Again, perhaps for another lecture, I can talk to you about what we're doing in terms of probing the brain for neurovisual diseases and different ways of treating depression, in fact. So with that, I will close off, I think, the lecture for today. And I would welcome your questions, and thank you very much for your time.
Okay, thank you very much to the speaker for a very interesting talk. So we do have a few questions from the chat in there right now. And again, I invite the audience, if you have any questions now at the end, to please post those in the chat, and we'll try to get them asked. So the first question that we have is, how much energy would there be in a typical pulse that would meet the cavitation threshold, and what frequency would you use?
The typical energies, let me come back to here, are on the order of tens of millijoules typically. Again, this is also dependent on whether you're trying to treat or dose. But if you want to stay at the threshold energies, you're talking about tens of millijoules per centimeter squared and that type of thing. And for the frequencies, especially on the detection side, the photoacoustic detection side, we're looking at on the ranges of hundreds or little less than 100 hertz pulse frequencies. Hopefully that answers the question.
Thank you. Next question from the chat is, for the photoacoustic imaging of the retina, are you using, or could you use the bubbles from the photomechanical cavitation as a photoacoustic contrast agent?
Well, in fact, I think that's exactly what we're doing. And I'm just going to come back to the other question here. The tens of millijoules that I'm talking about is really at the threshold range, right? So it does expand, the entire cavitation energies do extend longer than that. So I just want to mention that we're talking tens of millijoules for threshold energies. Regarding the cavitation and how we can use this as contrast, yeah, in fact, this, let me see if I can backtrack here. Daniel Pelenkar at Stanford has done beautiful time-lapse imaging here, and you can essentially look at the expansion and collapse of these vaporizations. But in the photoacoustic sense, that's exactly what we're doing. We're looking at that pressure wave that is generated by the microcavitation event, that vaporization event, and that pressure wave is exactly what we're detecting for, let's come back here, for this cavitation. So it is in fact, yes, the contrast agent.
Okay. So the next question refers to the triplet molecular oxygen that's created with the laser. And the question is if these are nanomolecules of triplet molecular oxygen, or if not, what is the scale?
Okay, I'm not sure I understand quite what the question is, but this is just simply taking molecular oxygen and, okay, so the photosensitizer itself are things like porphyrins, okay, they're porphyrin molecules, or phthalocyanines, and they're not large and they're not aggregated in any way. They're dissolved in aqueous solution, and the photosensitizer interacts with the molecular oxygen at the triplet state to produce singlet oxygen molecules, which are phototoxic. Those molecules, those individual molecules, are toxic to the tumor in which the photosensitizer is embedded in. And so it's a collection, shall we say, of the converted molecular oxygen from triplet state to singlet state that is responsible for the cytotoxicity. And in the case of where we're talking about radical species that are producing the toxicity, these are radicals and radical pairs, radical ion pairs that are responsible for the cytotoxic damage.
Okay, why don't we go on to the next question? What is the holy grail of laser energy confinement in the treatment and dosimetry of vision diseases?
Well, okay. So what do we want to ultimately do is be able to only target the lesion and not the healthy tissue. So I think the holy grail would really be in the ability to be able to confine this to one cell. There is a really there's a really interesting challenge in the molecular imaging world, the 1 millimeter tumor challenge. But if we can confine damage to only a single cell level, then we have a great deal of control in terms of the thermomechanical confinement to only that cell that we are interested in obliterating. So I think we're talking about nano precision surgery here. Well, let's call it cellular level precision laser surgery. I think that's where we're headed towards. In vivo. Okay, that becomes a little more tricky in diffuse media, right? We can do this probably a lot easier in non-diffuse media, but of course we have all sorts of photon migration that we need to consider. But single cell damage is where we want to get these types of techniques in terms of our selectivity and targeting.
Okay, and now I think we need to bring this to a close. So we'll ask the last question, which asks, how small is a single cell here in this?
How small is a single cell? Okay, it depends on the cells. Photoreceptors, 10 microns. They're longer. The rods and cones are different shapes and sizes, of course, rods and cones. But we're talking on the orders of 10 microns here. So we want to be able to confine that damage to, let's say, a single cell of that order of magnitude.
Okay, thank you. I think that brings the question session to a close. So again, thank you, Dr. Mermut, for the talk today. And with that, I would like to invite Bill Whelan to formally close out today's event.
All right. Perhaps I stop sharing at this point.
Okay, great. Thanks so much, Greg. At this time, I'd like to take this opportunity to thank Dr. Ozzy Mermut from York University for an excellent talk. Certainly illuminating from many perspectives, but some of the treatments for selective retinal treatments look very, very promising. And I'd say it's poised to have a huge impact on vision disorders problems. So congratulations for your work. I also want to thank our regional coordinator, Dr. Carl Adams from St. Francis Xavier University, who helped organize today's talk. Our host today, Greg Christian from St. Mary's University. Thank you, Greg. And a big thank you to Science Atlantic for sponsoring today's lecture. I'd like to take this opportunity also to thank from the CAP office, Francine Ford, Vicky Van Massenhoven, and Jenny Cleaver for all of their help in organizing the lecture series this year. And a big thank you to all of you for participating. This is the last talk in the 2022 CAP lecture series. I'd like to, as such, I'd like to take this opportunity to thank again all of the CEP regional coordinators, all of our speakers this year, and all of our host departments for delivering an excellent lecture series this year. So thank you very much to everyone again. And finally, a big thank you to the CAP Foundation for sponsoring the entire lecture series for 2022. As a reminder, as was mentioned earlier, all participating students are invited to remain to have informal discussions with Dr. Mermut in a Zoom breakout room. You should see a button with which to join the breakout room at the bottom of your screen shortly. So we encourage you to stick around. If you have any additional questions about the fascinating research Dr. Mermut is doing, or career paths, or just the whole area of biophotonics, I'm sure she'd be willing and happy to chat with you about that. So with that, I'll close out the 2022 CAP lecture series. I thank you all for participating and have a great day.